Okay. So, very good morning, my dear friends. Regarding of CPGET 2023 PG entrance online coaching and also all competitive exams JL, DL, Gurukul, JL, DL and PGT. This free online coaching, it is a wonderful platform for the young aspirants to achieve your goal. In this regard, today's our eminent resource person, Savita Madam Garu, Pulluri Savita Madam Garu, Assistant Professor, Head Department of Botany, GDC, Luxetipet. Madam, please welcome and share your screen and start your session, madam. Please welcome.
Thank you, sir. So today this is my third class regarding gymnosperms. We have dealt about the general characters of gymnosperms in our previous classes. Today I am going to deal with the classification of gymnosperms and also type study Pinus, life cycle of Pinus. Regarding classification, I think everybody is able to see the screen. Let me know. Are you able to see the screen? Okay. You come to the classification of the cycas. According to Spone, many scientists have given many classifications, but the most accepted one is according to Spone in divided the gymnosperms into three classes: the Cycadopsida, Coniferopsida, and Nitopsida. And under each class, 4 orders are there, and Nitopsida only 1 order is there. And when you come to Cycadopsida, 4 Pteridospermales, Benititales, Pentoxylales, and Cycadales. And Coniferopsida has Chordatales, Coniferales, Taxales, Ginkgoales, and Nitopsida has Nitales. So our cycas comes under Cycadopsida, and Pinus comes under Coniferopsida, and Nitum comes under Nitopsida. And mostly these fossil gymnosperms, they belong to Cycadopsida. So this is our classification. And in 1983, Stewart added another class, Progymnospermidae, where this consists of predecessors and ancestors of gymnosperms, predecessors or ancestors of gymnosperms. This is about the classification of gymnosperms. Next, when you come to the Pinus type study classification, it comes under the division Gymnosperm, class Coniferopsida, order Coniferales, family Pinaceae, and genus Pinus. So, family Pinaceae, genus Pinus, order Coniferales, class Coniferopsida, division Gymnosperms. So when you come to next, that is distribution of Pinus here. Pinus plants, they are distributed in temperate regions. They are distributed in temperate regions, and they grow in altitudes, in higher altitudes or slopes, in hilly areas. They grow mostly in hilly areas. They are evergreen, perennial, excurrent with cone-shaped canopy. They are evergreen. They appear green in color almost all the year. And when you come to their shape, the branches at the lower side, they are very long, and when you come to the apex, the branches will be very small, giving a cone-shaped structure. In India, they are distributed mainly in Himalayan ranges, Kashmir Valley, and hill stations of South India. Himalayan ranges, Kashmir Valley, hill stations of South India. So in India, we see 6 species of Pinus, that is Pinus longifolia or Pinus roxburghi, which is also called as chirpine, Pinus excelsa, which is also called as Pinus valiciana, or blue pine, or kale, or silver pine, and Pinus girardiana, which is also called as Silgoza pine, and Pinus insularis, which is also called as Pinus cassia or cassi pine, and Pinus nercusi and Pinus armando. This is about the distribution of the Pinus. Are you able to hear me? Are you able to hear me?
Yes, madam. Yes.
Okay. Next, we will go to the next slide. Morphology of Pinus. Pinus trees grows to height of 70 to 200 feet. They grow to 72, they are tall, and they grow to 70 to 200 feet. They are sporophytic in nature. They are diploid in nature. Means sporophytic is the dominant sporophytic stage, or the diploid stage is the dominant stage in the life cycle. And when you come to the root system, taproot system with ectomycorrhizal across this ectomycorrhizal association in Pinus. So where fungus associates with the external part of the root, and which helps in absorbing the water and substitute the function of the root hairs. This is about the root system. And when you come to the stem, Pinus bears two types of branches, long branches and dwarf branches. Long branches, they grow indefinitely and bear scaly leaves. Long branches grows indefinitely and bear scaly leaves at the nodes. And this in the axils of the scaly leaves, dwarf branches will arise and they bear cluster of needle-like leaves at their apex. And male cones are also formed in the in the axils of the long branches. Male cones are formed in the axils of the long branches. This is about the stem. Pinus bears two types of branches, long branches and dwarf branches. Long branches grow indefinitely and bear scaly leaves. And dwarf branches arise from the axils of the scaly leaves of long branches and they terminate in a cluster of needle-like leaves, which may be either 2 to 5 in nature. Next. Here you will be seeing the ectomycorrhizal association with the Pinus roots, and this is the Pinus branches, dwarf branches, where the branches are terminating into 2. On the main branch, dwarf branches are arising at the nodes in the axils of the scaly leaves, and they are terminating in a cluster of 2 to 5 needle-like leaves. This is about the Pinus stem. When you come to the leaves, two types of leaves are present, where scaly leaves and foliage leaves. Scaly leaves, they are membranous in structure. Please mute your phones. Please, students, mute your phones so that no disturbance can be seen. So these leaves are two types, scaly leaves and foliage leaves. Scaly leaves are brown in color and membranous, and they are present on the long branches. They are arranged spirally, and foliage leaves, they arise on the short branches, which end in 2 to 5 cylindrical needle-like structures. They appear triangular in cross section. These are persistent and fall only when the dwarf branches shed. When the dwarf branches fall, then only the needle-like leaves will be falling off. But otherwise, they will be living like that only. They are persistent in nature. Hence, these Pinus trees or the plants are evergreen in nature. Then when you come to the root anatomy, roots of Pinus shows, this is about the morphological external characters of the Pinus plant. When you come to the internal structure, root anatomy, roots of Pinus shows epidermis, cortex and stele. Epidermis, cortex, stele, and cortex and stele. These are the general characters of the anatomical features, general features anatomically. So when you epidermis, it is one cell thick parenchymatous in nature without intercellular spaces. It is a general character, one cell thick parenchymatous layer without intercellular spaces, and it is also called as piliferous layer because root hairs will be arising from the epidermis. Root hairs are present on the epidermis, hence it is also called as piliferous layer. When you come to the cortical portion next to the epidermis, the hypodermal, it is divided into hypodermis, general cortex and endodermis. But here hypodermal and hypodermal part and general cortex, they are not differentiated. They are parenchymatous in nature and they are not differentiated, and only the endodermis can be differentiated in the cortical region, which is brown in color and impregnated with tannin cells. So here in the cortex, you will be seeing that the hypodermis and general cortex are not differentiated. They are made up of parenchymatous cells, and the third layer, that is endodermis, it is differentiated, which is brown in color and impregnated with the tannin cells. So next, when you come to the stele, it is the outermost. So stele has three layers, that is outermost one is the pericycle, then vascular bundles, and centrally located medulla. Stele has outerly the outermost layer of the stele is the pericycle, which helps in the formation of the lateral root. Is the pericycle, which helps in the formation of the lateral roots. Stele is diarch to tetrarch in condition. It is diarch to tetrarch in condition. In this stele, you will be finding that the vascular bundles are xylem and phloem. Because it is root, radial vascular bundles will be present, which are arranged alternately. Here, radial vascular bundles are present, which are arranged alternately. Diarch to tetrarch means either 2 xylem bundles or 4 xylem bundles can be seen. And here, when you come to the xylem part, it is Y-shaped in structure with a protoxylem flanking the resin canal. Resin canal will be present where it is flanked with the protoxylem on either side, making a white. Metaxylem will be at the lower part, and on the either side of the resin canal, the protoxylem part will be present, which appears Y-shaped in structure. Xylem has only tracheids. Phloem alternates with the xylem and consists of seed tubes and phloem parenchyma. Vessels and companion cells are absent. Instead of companion cells, you will be seeing the albuminous cell. Phloem alternates with the xylem and consists of seed tubes and phloem parenchyma. Vessels and companion cells are absent in the phloem. The central part is parenchymatous medulla. So this is about the internal structure of the root. So root is divided into epidermis, the cortex, and stele. When you come to the epidermal part, it is single layered, thick, without any intercellular spaces and made up of parenchymatous layer. Because of the presence of the root hairs, it is also called as piliferous layer. Next is the cortical portion where hypodermis and general cortex are not differentiated, where endodermis is brown colored and impregnated with the tannin. And when you come to the stelar portion, it is made up of pericycle, vascular layer, which helps in the formation of the lateral roots. When you come to the vascular bundles, they are diarch to tetrarch in condition, where xylem is exarch and protoxylem is Y-shaped with resin ducts placed in the center. So xylem has tracheids. Phloem alternates with the xylem and consists of seed tubes and phloem parenchyma. Vessels and companion cells are absent here. So central part is parenchymatous medulla. When you come to the second, because of the presence of the here in the spinous root, you will be seeing the secondary growth. During the secondary growth, you will be seeing that meristematic tissue is formed between the xylem and phloem. Xylem and phloem are alternately arranged here because these are radial vascular bundles. From the portion between the xylem and phloem, meristematic tissue arises, and which performs the function of the cambium and cuts of xylem inside and phloem outside. From pericycle, you will be finding cork cambium is formed, which cuts cork towards outside and parenchymatous secondary cortex inside. Cork cambium and secondary cortex are called as phelloderm. This is about the secondary growth in the root. Here you can see in the diagram, this is the needle-like structure. These are the first diagram, which shows needles, that is the dwarf branch, which is terminating into needle-like structures. Second is the pinus plant, and third one is the corolloid roots of the pinus. And for the internal structure of the root, where outer one is the epidermis, outer layer with single-celled thick layer, which has the root hairs. And next is the parenchymatous cortical region, and inner one is the endodermis. And when you come to the stelar portion, the outer layer is the pericycle, which has here phloem and xylem. In the xylem, you are seeing the and medulla is present in between. This is about the internal structure of the root of the pinus. When you come to the anatomy of stem of pinus, it shows three regions as you have seen in the root. There are epidermis, cortex, and vascular cylinder. Epidermis, it is one cell thick parenchymatous layer covered with the cuticle. Hypodermis, middle general cortex, inner endodermis. Epidermis, it is parenchymatous and made up of one cell thick layer without any intercellular spaces, and it is covered by cuticle. And cortex divided into outer hypodermis, middle general cortex, and inner endodermis. Hypodermis is sclerenchymatous and 2 to 3 layered in structure. Hypodermis is sclerenchymatous and 2 to 3 layered. And when you come to the general cortex, it is parenchymatous and possesses numerous resin canals, which is an important characteristic feature of the pinus. In pinus, you will be seeing in the resin canals are distributed in many parts of the plant, in the stem, leaf, etc. You'll be seeing the presence of the resin canals. These resin canals, they are schizogenous in nature with epithelial lining. Schizogenous, they are formed by but as the cells move apart, a cavity is formed, and this cavity is covered by epithelial lining. These epithelial cells secrete turpentine into the resin canal, which is stored in the resin canals, which has an economic importance. This turpentine secreted by the pinus has economic importance. It is used for varnishing, it is used in making paints, etc. Innermost layer is endodermis without any Casparian strips. This is a characteristic feature in the pinus where the endodermis is not having any Casparian strips. Casparian strips are nothing but the lignin or suberin bands present on the endodermis, which were discovered by Casparian and scientists. These were discovered by Casparian. These are named as Casparian strips. So this is about the anatomy of the stem. So it shows three regions: epidermis, cortex, vascular cylinder. And epidermis, it is one cell thick parenchymatous in nature. Cortex divided into outer hypodermis, middle general cortex, and inner endodermis. Hypodermis is sclerenchymatous and 2 to 3 layered in structure. General cortex is parenchymatous and consists of numerous resin canals, which is a characteristic feature of the pinus. These resin canals are schizogenous in nature with epithelial lining which secrete turpentine into the resin canals. Innermost layer is endodermis without any Casparian strips. So here you can see the diagram. Outermost layer is the epidermis, which is flanked by the cuticle, and next to the hypodermis, sclerenchymatous hypodermis will be there. So where general cortex, you are seeing the resin canals. Many resin canals are present in the cortical region, and endodermis, vascular bundles are arranged in a whorl-like fashion, which resemble the vascular bundles in the dicot plants. So this is the diagram of the TS of pinus stem. So when you come to the vascular bundles, they are conjoint collateral, and because of the presence of the cambium in between, they undergo secondary growth. The xylem consists of tracheids only, whereas phloem consists of phloem parenchyma, phloem fibers. Their companion cells are absent. Instead, albumin cells are present in the phloem, and in between the xylem and phloem, you will be finding the xylem rays or the medullary rays, where in between, in the central part, medulla is present, which is parenchymatous in nature. So next, when you come to the secondary growth in the stem, secondary growth in the pinus resembles dicot stem. This parenchymatous cells in between the vascular bundles transform into interfascicular cambium, connecting the intrafascicular cambium. The cambium present in between the xylem and phloem in the primary stages is called as the intrafascicular cambium. In the primary structure of the vascular bundles, you will be seeing a cambium, a strip of cambium will be present in between the xylem and phloem. which is called as intrafascicular cambium. This during the secondary growth, a cambium is formed in between the two vascular bundles from the meristematic tissue present in between the two vascular bundles, which is called as interfascicular cambium, which connects the intrafascicular cambium, forming a continuous cambial ring. This cambial ring cuts off new xylem towards inside and flows towards outside. This cambial ring cuts off new xylem towards inside and phloem towards outside. The secondary xylem is pycnoxylic in nature in Pinus. For the purpose of entrance examinations, secondary xylem is pycnoxylic in Pinus. Annual rings are formed during the secondary growth. Annual rings are with distinct springwood and atom wood. Means during the springwood, you will be seeing that the vessels will be large in structure, and during the atom, the vessels will be these tracheids, the tubes will be tracheids will be very large, and during the atom wood they are small in nature, because during the atom you will be seeing that the leaves will be shed off. So in spring season, the leaves will be the young leaves will be forming at that time, hence they need more water, hence the tracheids in springwood will be very large in structure, and during the atom wood they are small in nature, forming two tracheid layers which make one annual ring. So annual rings are formed with distinct springwood and atom wood. When you count the number of the annual rings, you can know the age of the pinus tree, which is called as dendrochronology. So biseriate pits are present in opposite manner on the secondary xylem, and which are called as Abitanian pits. You will be finding on the secondary xylem, pits are present. Biseriate pits means double-layered pits are present, which are called as Abitanian pits. And in between these pits, you will be finding the bars of Sanyo, which are also called as crassulae. These are the important characteristic features of the pinus. So secondary phloem consists of sieve tubes, phloem parenchyma, phloem fibers, and no companion cells are seen. Instead of companion cells, you will be finding the albuminous cells. Xylem and phloem rays are formed from the cambial cells. Secondary growth in the cortical region leads to the formation of the cork cambium, which cuts off cork outside and secondary cortex inside. This is about the secondary growth. So it resembles the dicot stem, where interfascicular cambium forms between the connecting the intrafascicular cambium, which forms a continuous cambial ring, which cuts off new xylem towards inside and phloem towards outside. Here the xylem formed is pycnoxylic in nature. You will be seeing that annual rings are formed, and which helps in the counting the age of the pinus tree, which is called as dendrochronology. To know the age of the plant by counting the annual rings is called as dendrochronology. Here biseriate pits are present on the secondary xylem and are called as Abitanian pits, and bars of Sanyo are present between the pits, and these are also called as crassulae. Excuse me. Secondary phloem consists of sieve tubes, phloem parenchyma, phloem fibers, and companion cells. Xylem and phloem rays are formed from the cambial cells. And secondary growth in the cortical region leads to the formation of the cork cambium outside and secondary cortex inside. When you come to the anatomy of the pinus needle, about the leaf, in cross section we see that needle appears triangular in shape. Needle appears triangular in shape, which has outer epidermis, with a thick cuticle, and here epidermis has sunken stomata, which is a xerophytic nature. Sunken stomata are present in the epidermis, and below the epidermis, 2 to 3 layered hypodermis is present, which is clearing cametus in nature, which is also a xerophytic character. Here in pinus, you will be finding many xerophytic characters. In the leaf, in the leaf part, you will be finding many xerophytic characters. One is the presence of a thick cuticle, and second is the presence of this hypodermis, which is clearing cametus in nature, and sunken stomata are seen, And these sunken stomata form a stomatal chamber in the sclerenchymatous hypodermis. And here the leaf is divided into three parts: epidermis, mesophyll, and vascular bundles, or the stellar portion. So below the epidermis, you are seeing the hypodermis, which is sclerenchymatous in nature. And next to the epidermis, hypodermis, the mesophyll tissue is there, which is not differentiated into spongy and parasite parenchyma. Mesophyll contains parenchymatous cells, which are not differentiated into spongy and parasite parenchymatous cells. The walls of the mesophyll cells, they are projected into finger-like structures to increase the surface area of the leaf. They are called as armed parenchyma. The walls of the mesophyll cells, they have finger-like projections which increase the surface area, and these are called as the armed parenchyma. Mesophyll cells also contain resin canals with epithelial lining, as if you have seen in the stem part, secreting turpentine. Endodermis is prominent with barrel-shaped parenchymatous cells and Casparian strips. Endodermis is here, it is clearly visible, and it has barrel-shaped cells which are parenchymatous in nature and have Casparian strips. Yeah, when you come to the vascular cylinder, it consists of transfusion tissue between the endodermis and vascular bundles. Transfusion tissue between endodermis and vascular bundles. As we have already told in our learned in our general characters that these gymnosperms, they have only midrib in the leaf, and lateral veins are absent. To substitute the function of the lateral veins, here a tissue called as transfusion tissue is present between the endodermis and vascular bundles, and it carries out the function of the lateral veins. This transfusion tissue has four kinds of cells. It is present around the endodermis. It runs parallelly along with the endodermis up to the root portion. So here the transfusion tissue has four kinds of cells there: parenchymatous cells, albuminous cells, transfusion truncated cells, and sclerenchyma. So transfusion tissue has four kinds of cells: parenchymatous cells, albuminous cells, transfusion truncated cells, and sclerenchyma. The parenchymatous cells, they help in the transport of the water from vascular bundles to mesophyll. So near the cilium tubes, parenchymatous cells store starch and are called as albuminous cells. Here, cilium parenchymatous cells store starch and are called, whatever parenchyma is associated with the transfusion tissue, they carry on the function of the store the starch, which are also called as albumin cells. And xylem has thick lignified dead cells with bordered pits, which are called as transfusion truncates. Lignified dead cells with bordered pits, which are called as transfusion truncate cells. T-shaped sclerenchymatous tissues present between the two vascular bundles. T-shaped sclerenchymatous tissues present between the two vascular bundles. There are two vascular bundles present in the central part of the leaf. These vascular bundles are conjoined, collateral, open, and endark in nature. Conjoined, are present side by side, collateral, they are present on the same radii, and open, they have the cambium in between, and endark in nature. So where the protoxylem is towards the medulla side. So here two vascular bundles are seen. In between the two vascular bundles, sclerenchymatous tissue is present, T-shaped sclerenchymatous tissue, which is associated with the transfusion tissue. So sieve tubes, phloem parenchyma are present, whereas companion cells are absent, and companion cells are substituted by albuminous cells, which perform the function of storing the proteins. This is about the anatomical structure of the leaf. Here the leaf is needle-like. The needle-like leaf is which it shows triangular nature. In cross section, you see the triangular nature of the needle, and the outer part is the epidermis, which is thick and which is covered by the thick cuticle again, and which has sunken stomata. And below the epidermis, you will be finding the hypodermis, which is sclerenchymatous in nature. And below the hypodermis, Mesophyll is made up of parenchymatous tissue. Here in the diagram, you seeing that finger-like projections are present, which are called as armed parenchyma in the mesophyll cells. Here, no differentiation of palisade and spongy parenchyma is seen. This armed parenchyma helping increasing the surface area of the leaf. And when you come to the central part, that is the resin canals are also present in the mesophyll cells. And when you come to the central part, that is the vascular cylinder, which has endodermis, which has Casparian strips, and transfusion tissue is present below the endodermis. Between the vascular bundles and endodermis, you will be seeing the transfusion tissue, which run all along the endodermal part. So here you will see in the transfusion tissue consists of 4 types of cells: that transfusion tracheids, sclerenchyma, transfusion tracheids, sclerenchymatous cells, and you will be seeing that albumin's parenchymatous cells. The parenchymatous cells help in transportation of water and storage of the starch, and transfusion tracheids are present in the xylem, and T-shaped sclerenchymatous cells are present in between the vascular bundles. 2 vascular bundles are present in the central part, which are conjoined, collateral, open, and endarc. And here C tubes will be having the C tubes, phloem parenchyma are present in the phloem, where companion cells are absent and they are substituted by albuminous cells. When you come to the xerophytic characters of the pinus, presence of needle-like leaves, presence of thick cuticle, sunken stomata, sclerenchymatous hypodermis, presence of transfusion tissue. This is all about the characters of the morphological characters and anatomical characters of the pinus. If anyone has doubt regarding any topic, any of the part in this today's topic, you can ask me.
Any doubt, my dear friends? Madam has clearly explained about the complete detailed about the morphological and anatomical characteristic features of the pinus of the gymnosperms, my dear friends. Regarding of today's class, have you any doubt? Please you ask our respected resource person. Okay. Thank you, madam. Nobody has raised any doubt. So you are clearly explained about the total information regarding of this morphological and anatomical features. Thank you, madam. So
Thank
tomorrow,
you,
or day after tomorrow, maybe tomorrow.
Day after tomorrow, we will be dealing with the reproduction in cycle in pinus. So thank you one and all for listening patiently with your extreme patience.
Okay, madam. Thank you, madam. Thank you very much.
Okay, sir.
Right, madam. Right.
Okay.